Hello, everyone. Welcome to today's live broadcast, Biomarker Discovery, Metabolomics Differentiates Known Disease Classifications of Prostate Cancer. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by SCIEX. SCIEX helps to improve the world we live in by enabling scientists and laboratory analysts to find answers to the complex analytical challenges they face. To learn more about SCIEX, please visit sciex.com. We have a few important announcements before we begin. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button at lower left. Before we meet today's speaker, I want to advise that a series of poll questions will appear on your screen. You may select your answers, then close the poll questions by clicking on the X in the right corner. The first question is, what are your current metabolomics workflows? And the choices are A, untargeted, B, targeted, C, both, and D, not performing analysis. And the next question is, are you aware of swath acquisition workflows for metabolomics? A, yes, B, no. And the last question is, what is your biggest challenge with these workflows? And please choose only one response. A is shortage of skilled personnel, B, lack of standardized protocols, C, data complexity, D, availability of metabolomics libraries, E, other. Thank you. I would now like to introduce today's speaker, Tim Garrett, PhD, Associate Professor and Director of High-Throughput Metabolomics for the Southeast Center for Integrated Metabolomics. Dr. Garrett earned a PhD in Analytical Chemistry and later joined the faculty in the Department of Medicine at the University of Florida as a Research Assistant Professor and Core Laboratory Director for the General Clinical Research Center. In 2014, he joined the Department of Pathology, Immunology and Laboratory Medicine as an assistant professor. Dr. Garrett's research is focused on the application and development of mass spectrometry techniques and instruments for clinical research and translation to clinical diagnostics. He has a specific interest in developing approaches to small molecule quantitation or characterization using mass spectrometry to improve our understanding of diseases, drug treatments, and therefore health. This interest involves the use and development of imaging mass spectrometry, IMS, to directly probe excised tissue sections for chemical distributions, traditional liquid chromatography, tandem mass spectrometry of homogenized tissue and plasma or serum, and high-resolution mass spectrometry for profiling the human metabolome. He has been at the forefront of developing instrumentation and applications for IMS studies and plans to continue developing this technique to provide high-throughput analyses and better identification of unknowns. I will now turn it over to Dr. Garrett for his presentation.
Great. Thank you very much for that introduction, and thank you for this opportunity to talk today. And I also want to thank the audience for logging in to spend the time. And this is a unique opportunity for me to describe some of our work that we're doing, and specifically on the use of metabolomics in understanding prostate cancer and understanding the health of the prostate. So first I'll begin by talking a little bit about metabolomics at the University of Florida, and specifically our center, the Southeast Center for Integrated Metabolomics. We have 6 cores that help conduct the work that we do, an administrative core that oversees the operation, a promotion and outreach core, P&O core, that enables new researchers access to metabolomics, and an informatics and tools core that's building new tools to understand how to dig information out of your data. And of course we have an analytical side that includes nuclear magnetic resonance for quantitative metabolomics, advanced mass spectrometry that includes the imaging mass spectrometry capability. And then, of course, my core, which is liquid chromatography high resolution and tandem mass spectrometry, in which we do multiple kinds of approaches to probe the metabolome. And information from our center is often uploaded to Metabolomics Workbench, which is an NIH-funded website. We, of course, are an NIH-funded center, one of the 6 centers that is funded through the NIH Common Fund. And you can get more information from the Metabolomics Workbench, including standard operating procedures, and you can download data sets that have been uploaded for multiple resources. Some of the services that we offer, of course, are global metabolomics, which is profiling hundreds to thousands of metabolites in a single sample, as well as lipidomics, which looks at the more non-polar part of the metabolome, including a variety of classes of lipids that you'd want to detect. And of course, targeted quantitation. And targeted quantitation includes many different assays, but some of the examples are acyl-carnitines, organic acids, amino acids, and pyridine nucleotides. And some new ones are measuring 1-carbon metabolism, as well as mass spectrometric measurement for DNA methylation. As part of our center, we offer tools and workshops. We have an annual workshop to teach people metabolomics. We use our website to deploy new technologies, ways in which you can dig into the data, and some of the things I mentioned earlier about funding projects. And of course, a key part of this is encouraging new faculty to use metabolomics and trying to teach them the best opportunities that are there. A key part of any metabolomics is being able to identify things, and so we have specific tools that we've used in the past and that we're developing to be able to identify new and unknown metabolomes. AROA is one of those, which is isotopic ratio outlier analysis, that involves both tissue analysis using flux and fluxomics approaches to follow pathways to find out where metabolites are going, and then combining some NMR capabilities to really do structural elucidation and identify what that unknown is. So if you're not familiar with metabolomics, I'll give you a short overview of it. Metabolomics covers a wide array of things that are in our body, including the genetics that is a part of us. Our age determines some of what our metabolome looks like. Where we live, the environment, what we ingest, all goes into our metabolome. Of course, if we have an illness or if we're not feeling well, those have effects on our body, and those are often seen in the metabolome. If you're taking a pharmaceutical or over-the-counter drug, you'll see metabolites from that drug. That's part of the exposome. And nutrition, as I mentioned, is part of the environment, but it's also a sort of a separate category because there's many different things that are included in nutrition, and that, of course, will change with age as well. And your lifestyle, your exercise or sleep habits, all of those affect your metabolome, and that then translates to your phenotype. So part of metabolomics is to understand all those factors that go together, which is difficult, but can be deciphered using different and various approaches. A key part to say is that we are diverse, right, because we all do different things, but in the end we can come to a conclusion on some types of phenotypes that would be similar by measuring enough samples. In our research example, I'm going to talk to you today about the analysis of urine for prostate cancer. Urine, of course, is the second most common cancer among men. It's a very high treatment success rate, but there's also overtreatment, and there's not 100% success in terms of diagnostic potential. PSA tests are often done as well as following DREs to do early detection, and PSA is not really reliable at times. It can lead to overtreatment and has led to overtreatment in the past, and screening doesn't often correlate to the stage of the cancer. And so our question as a part of this study is, can we distinguish individuals with prostate cancer using metabolomics by screening the urine? And part of this also is understanding whether or not urinary metabolomics will be useful as a future clinical diagnostic tool for them. An interesting part when you look at the history of science and you look at metabolomics, we've been doing metabolomics for a long time, since the 1500s with the urine wheel that was published, primarily looking at the smell, taste, and other factors that are involved our senses to understand what was in the urine, and this would be related to diabetes or other things that might change different aspects of your urine. So urine was really the first metabolome. The good thing about it is that we have really nice analytical tools today, besides our noses, so that we can probe the urine in an exquisite way and really look at chemical structure and understand what's there. And this is figure that I show is just using NMR to look at urinary metabolome. So one thing that's also been done in the past is looking at the liquid biopsies. And so this gets at the fact that urine is a good clinical diagnostic paradigm. Urine is important for diagnostics. It's non-invasive. It's very easy to collect. You can get a large volume if you need to concentrate it, and so that allows you to do different kinds of studies that might require different volumes. And that would be exosomes. So exosomes can be collected. These are vesicles that are excreted. You can get RNA, and of course you can get metabolites, and metabolites will be correlated with disease. And recently, the exosomes have been sort of an explosion. A lot of interest, even though they were discovered several years ago. The interest has gained momentum more recently because of the ability to measure what's inside those liquid biopsies and what's inside the exosomes as transmission or metabolites or other things, proteins that might be contained in there that tell you what's going on inside the body and what's being transmitted between different organs. And so looking at those kinds of things in the urine is a very important opportunity for clinical diagnostics. And there's recent companies that have done this. This is one example. There's other ones that are looking into this area to do diagnostic opportunities. So what we're focusing in on is, of course, the small molecule part of this, those that are excreted out. And there's two different opportunities in metabolomics. If you look at that, you have a discovery on the top part of the slide that basically discovery means that you don't really know exactly what you're looking for, but you're looking for many different opportunities and metabolites that might show you what is changing within the system. And that's usually employing a high-resolution system, looking at thousands of features over multiple samples and using high-level statistics to pull those out. And the other opportunity is looking at a targeted set of assays or a targeted set of metabolites, say maybe a smaller set of metabolites, 10 to 100 or so, and that allows you to identify and quantitate. And you can do those on the same platform or on different platforms. Because of the capabilities of this platform that we're using, you can do both MS, and MS on the same sample with the same injection. So you can sort of get a comparative study to look at targeted opportunities as well as untargeted opportunities. And typically the flow is once you move to a targeted assay, you increase the numbers of samples that you're running because you're focusing in on a smaller set as you go forward. Our workflow for doing global metabolomics of urine is collecting the urine. We measure the specific gravity. We have other measures that are clinically often used that we can correlate to other components. But for this case, the specific gravity is a key part in sort of looking at the normalization between samples. In any study, we have to add internal standards. And so a key part of it is following the process, making it very controlled, and looking across the samples with the same standards, making sure you get the same response. And in this case, we have the internal standards, and then we added acetylnitrile, and then we spin down and dry it and reconstitute it in water. And those steps are really to get rid of the sediment and to try to get a cleaner sample out. And the analysis is done by electrospray ionization in both positive and negative ionization on a X500R, with separation on a C18 PSP. This is a polar embedded stationary phase at a typical flow rate for high flow metabolomics, 350 microliters a minute, with a turnaround of runtime between 22 minutes to 23 minutes from injection to injection. And then the final step of that is running these under the conditions. Usually we bracket these. We run these as subjects as 10 groups and blanks and controls and run through a set of samples to get an idea of this. And then we do statistical analysis, aligning all the features. And so if you look at that, this is an example using the software marker view to look at the variation of what's present. And on the left side here, we're up at the arrow. This is what we're looking at in terms of the principal components analysis, showing the separation of the various types of samples we have. And then on the right side would be the components that are the scores plot that are that are showing what's in that separation. And so if we highlight the yellow, those are the controls that we collected. That's just a separate set of controls that allows us to monitor the quality of the sample, so really more quality control. Then we have a separate set of QCs. These are neat QCs that we add to every sample. Neat QCs just provide a smaller subset of metabolites that we can measure and routinely check to make sure retention time and mass accuracy are where we expect them to be. Then we have where our clinical study, where we have our healthy subjects that are clustering very close together, and then we have those that have prostate cancer that are clustering up at the top. So this in the beginning shows us the ability to separate the prostate cancer subjects from those that are healthy, which is where we wanted to. So in the beginning, we're now seeing the ability to use just metabolomics without looking at which metabolites are there, but using the global expression to separate one group from the next, and then getting good quality measures using two separate kinds of controls. You can then pull out specific information, and you can pull out the masses and the retention times of those masses to correlate which metabolites are expressed in which one. So on the left, you're going to see those that have prostate cancer, examples of metabolites that are differently expressed in prostate cancer versus those on the top that are healthy. And of course, you can use other statistical approaches. We like to use other statistical engines to do different validation studies, and so we often export the data from the software to then go do additional statistical analysis in MetaboAnalyst or other tools that we have at our disposal to do further validation studies. So, metabolomics in prostate health. Well, cancer, of course, is important to distinguish, right? But if we're trying to do a new diagnostic potential, we have to understand the health continuum. And that is important because we have a subject coming in that we don't know whether or not they have a disease. And so other manifestations of health would be patients who have prostatitis or those that have BPH, which are more related to inflammation rather than a cancer. And so this is an important component to do and conclude in your validation studies to know that you can separate a variation of health rather than just separating cancer from normal. And so this principal components analysis on the right shows a grouping of three different groups. And I'm going to highlight again, we have a normal group, and this was done with MetaboAnalyst, an R-based system. And on the right, we have the separation of our prostate cancer subjects, just as you saw before in the previous plot. But in the middle, now that we add those that have inflammation, those with prostatitis and BPH, we can see that they tend to cluster together. So we can't separate individuals who have one or the other, but it does appear that we can separate those that are not healthy but don't have prostate cancer, which is a very important component that we want to have in any kind of clinical study. And then we can pull out individual expression. And this is really where you're finally getting into the information that's in the metabolome, and you can look at changes associated with those different groupings. So we can look at alanine and sarcosine. In our platform, we don't separate those. In the subsequent targeted assay, we will validate the separation of alanine and sarcosine to determine which one is causing the separation. But in the end, you can sort of see that the normal subjects have a low expression of alanine and sarcosine, while those that have cancer or BPH or prostatitis are high. But you can also point out here that both with that one and with creatine, that there's no real difference with those with cancer and prostatitis or BPH. And this is an important thing to raise, in that this is why we need more than just one manifestation of disease, because we need to know what the expression looks like for an overall health versus just two groups. And then if you look at glutamine on the right-hand box plot, you can sort of see what that looks like now that you have both components together. You have those that have BPH and prostatitis at a separate level, those that are normal having a high expression of glutamine, and then those subjects with cancer having a low expression of glutamine. And some of the previous work has shown that glutamine is essential for tumor growth, and lower levels may be indicative of that, but of course that requires further validation. There was a study that looked at sarcosine being increased in prostate cancer tissue. Remember, this is urine, so this expression of sarcosine in our urine, we don't know how that correlates to the tissue example, but there is previous work that shows that sarcosine and glutamine may be important factors to look at when you go forward with this kind of diagnostic opportunity. So you're starting getting more of a visual picture. And as was mentioned earlier, I do imaging mass spectrometry. It's also metabolomics, and I think an image tells you many different things and can be easier to express. And so we have to create a better image of health to show us what the spectrum of health really looks like. And so here we're starting to see a better expression of that health. Okay, so there are opportunities to do targeted metabolomics at the beginning versus doing global. I like to start with global metabolomics. But we did in this study do both because you get different information. So why not just start with targeted metabolomics then, where you can get quantitative information and you get that information readily. So we can run a set of well-known clinical assays and we can get that information. And so in this case, we ran organic acids, isocarnitines, and amino acids. And from this work, if we do the same sort of principal components analysis, we don't see the clustering. We do notice that we can distinguish those that have cancer from those that were normal, the dark blue versus those that have the light green, and that maybe not as clean as we had with global metabolomics, but we can't separate those that have prostatitis and BPH from that group. It looks much more scattered. It is important to note that individually there are many significant metabolites within that quantitative run, as you would expect. But as a whole, they don't describe that spectrum of health as well as what we showed before. And I'll put that plot back up there for untargeted. So with untargeted, if you put these two together, we can improve our potential ability to distinguish this kind of health paradigm. And this really means there's expression beyond what we normally run that allows us to see more information. And this is one of the reasons doing an untargeted approach may enable new clinical diagnostic paradigms. So the metabolome is many different things, right? And there's unknowns, right? So we measured knowns, but in our data set, we also have things that we don't know what they are. And that brings up a point raised about what is in the metabolome and what is not. And so there's a lot of future research going on to deciphering more metabolites that are present within the pathways. The CAG pathways were developed many years ago, and of course we're seen as very, very important. And progress was made in the 60s to make these a validated step. And then more recently, more in 2012, basically with our understanding of metabolism, it continues to evolve and continues to grow. And so this represents an opportunity in metabolomics to jump into this space and to do new discovery that helps understand it. And part of this discovery involves identifying things, right? And so you need opportunities and you need tools to identify what's there. And so this is an example that might be there with Formula Finder and ChemSipder database searching. Getting the right kind of formula is important, and that involves getting the isotopic pattern. And so what you can see here is the ability to both look at the extracted ion chromatogram, the XIC of an individual sample, and that would be here. So extracting a sample out, looking at its expression. But the way we collected this information was we were able to collect in both full scan, and so full scan would be right here. So collecting the full scan information, this is under high resolution to get our isotopic pattern. And then the final step would be actually to get MS under swath. And putting all of this together allows you to identify potential unknowns for further validation. You need high mass accuracy to get the right mass and to distinguish it from other noise. And you need MS to help you understand how it breaks down to match against databases that have already been done. And so this is an example of looking at MS1 from formula ID, looking at the structure information and then getting an MS2 match for that specific compound. And this is an example for phenylalanine. And so the key part here is getting a match of the structure, getting the mass of the fragment ions under MS2 to see how those correlate with the rest of the structure, and then getting hits that tells you how those fragments match up with a theoretical distribution And this allows you to finally start digging into things that you don't know, because you can use those fragmentation patterns to make connections between other potential either known metabolites and to start building a set of unknown fragmentation patterns and build a new structure, or confirm those that are present that are already known. And a thing about SWOT to note is that it's an important component and an important feature that allows you to do MSMS in a different way, because you can isolate a window that's very adjustable. So you can do a large or small window up to 50 Daltons or down to 10. And one reason I like this component is that when we add an internal standard, and if you want to look at isotopic ratio outlier analysis, in that approach, we add an internal standard that is the exact number of carbons larger than the carbon-12 side. And so we need a larger window to isolate both the carbon-12 and the carbon-13 side. So why would that be important? For us, it's important because it allows us to follow the fragmentation pattern and see what's falling off and be able to know that we've lost one carbon or we lost two carbons and build that information together. It's very important for unknowns in that regard. And having this flexible window allows us to put those two in the same swath window and collect that information downstream and then do post-processing to analysis. An example that's shown here is looking at fructose bisphosphate, which allows us to look at the different fragments and how those fragments match up with each other to help understand which fragments go together with which parent. And it's important with that because swath collects a larger window. You need to match which ions go with which parent, and that's a very needed assessment and needed tool in metabolomics. Okay, with that, I hope that I've given you a expression and an idea of where metabolomics can go in the future and what it can do for prostate cancer diagnostics. I've shown you the ability of untargeted workflows to decipher the spectrum of health and the spectrum that's within the prostate cancer diagnostic potential for future. And we hope that under our current guidelines and our current research that we'll be able to expand in the near future for a larger scale study to be able to confirm that this is true. And so with that, I'd like to thank those that have worked with me on that and on this study, including Dr. Ubi from SciEx, collaborator Ranjan Pereira from the Sanford Burnham Institute in Florida, a visiting scientist from Poland, Paul Derezinski, who helped analysis a lot of these in this project, and Vanessa Rubio, who is a graduate student in the Department of Chemistry currently working on it. And of course, I'd like to thank the funding sources and my collaborator, SciEx, for working together with me on this. And finally, I'd like to thank you for your attention and willing to answer any questions that you may have. Thank you very much.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, how do you normalize the data given that volume output will differ person to person?
So it's an important thing in urinary metabolomics. We normalize to specific gravity, and that allows for variation in what's present in terms of the salt concentration. We have found that normalizing to creatinine, and others have found the same thing, that it does not work as well in a global metabolomics run. Another published report talked about the use of using the total useful signal as a way to normalize the expression across different samples. And in this study, we focused on specific gravity as a way to do that, and it worked fairly well. And under validation conditions, it seemed to show the right expression at the right time.
Did you collect the MS and MSMS in separate injections, or did this platform allow you to collect the data together?
This platform allowed us to collect the data in the same injection, so that enables us to use less sample or to save the sample for other things in the future, and then to use the information post-processing to dig out to identify and dig out other information that's present. And that also allows you to have less injection so that you can keep your instrument running on multiple projects down in the future and improves our overall performance.
What acquisition mode did you collect this data, data-dependent or data-independent?
This was collected in a data-independent mode so that we could look at things that we don't yet know what's there and that we could gather as much information as possible.
Next person says, very interesting data. What are the follow steps to this pilot study, and is there a larger validation study planned?
Our follow-up steps have been in two different areas. One, we validated this on multiple instrument platforms, and we've also validated it with multiple separation modes, including reverse phase and Hillick. And our next phase is working with clinicians and other biorepositories to gain access to new samples to expand this to hundreds of samples rather than a small subset of 40 that we had across this study. So hopefully in the next year or so we'll complete those final steps and have a validation study to be published and presented to the world.
I would like to once again thank Dr. Garrett for his presentation. Do you have any final comments?
I have only one comment, and that's to say that I think that our future with metabolomics has a high potential. There's a lot of things that we don't yet know exist, and I think that more scientists jumping into this field and applying their scientific knowledge and understanding of metabolism will aid in future diagnostics. And I hope to see that you'll begin studies in metabolomics if you haven't already done so.
Well, thanks again. I would also like to thank our sponsor, SciEx, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available on demand through April of 2018. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time.